beginning. We're very excited. We have a really good panel today and a very capable moderator. So I will now hand over to Hannah Lester, who will moderate today's webinar. And thank you very much for your attention.
Super.
Hannah,
Thanks,
over
Lucy.
to you.
Thank you so much, Lucy. So good afternoon, everyone, and thank you for joining us today, where we're going to be talking about how novel foods get to your plate. We've got a wonderful set of speakers who in turn will walk us through the pathway to market for novel foods from initial R&D through to the novel food risk assessment, approval, and consumer acceptance. So to kick things off, when we're talking about novel foods, we mean foods or a food ingredient without a significant history of safe consumption within the European Union prior to the 15th of May, 1997. So I work with companies of all sizes that are looking to get their novel foods to market in the EU. And this really is a huge task. And it's really an interdisciplinary and multi-year effort. And for some novel foods, such as plant proteins and human milk oligosaccharides, we have precedents. So as applicants, we know which studies and data are required to support safety, and we know roughly how long it's going to take from submission to approval, as long as we follow the guidelines and prepare a good quality dossier. For novel foods where there are no precedents, such as cultivated meat and seafood, the pathway is more uncertain, as the data requirements are not always clear. And also the authorities are not familiar with the technology, and so it's going to be the first time that they're performing the risk assessment, and the risk assessment is going to be new to them. To help applicants prepare, EFSA has published guidance documents, and today is a very exciting day as the updated guidance for novel foods, traditional foods, and the bioavailability of new micronutrient sources were published today. So I'm sure Umalaus is going to give us an update on these very important documents. And for all novel foods, bringing them to market requires a significant investment of time and money. And it's also a real challenge to translate a product from R&D into a novel food that's produced at commercial scale in compliance with the relevant legislation. At R&D stage, it's important to start thinking about regulatory feasibility as early in your journey as possible, as this can really help to inform your product development strategy. And for example, for products that are produced using microorganisms, the type and the species of microorganism that you select has implications in terms of the studies and data that you'll need to provide. And if you're working on cultivated meat and fish, do you have all of the relevant information and data on the source of your cells, and also traceability in terms of what the cells have been exposed to during isolation, adaptation, and banking? And when you're ready to start producing your regulatory batches to support your novel food dossier, which studies are required? Also, importantly, which studies do you need to notify to EFSA prior to the study start date in order to be compliant with the transparency regulation? And when you submit your dossier, what is the process and what are the timelines? EFSA may conclude that your product is safe, but then what happens during the risk management phase, and how are novel foods approved? And even if they are approved, are consumers going to want to eat these novel foods? So throughout today's talk, we're going to do a deep dive into each step of this process, and you'll learn about the scientific evidence required to support novel foods, how novel foods are assessed, and what the future holds in terms of food innovation. And by the end, we hope you'll have a clear understanding of all the steps it takes to get your novel food to your plates, making sure that it's both safe and nutritious. So thank you again for joining us. And I'm delighted to introduce our first speaker. So we have Per Falholt, who's the CSO from 21st Bio. And Per is going to be talking about the research and development of novel foods. So over to you, Per.
Yes, thank you very much, Hannah. And my point here is that I want to give you a little bit of update on what we have been doing in order to develop new proteins for human consumption. And of course, you can discuss to a certain extent how new are they, but I think for regulatory purpose, that is a definition we need to work on. Our experience with new proteins is right now based on a protein called beta-lactoglobulin. It's present in milk, so it's a protein we have been consumed for thousands of years, and we are producing it using a microbial host, which have been approved for at least 50 food products over the last 60, 70 years. So the strain background is very, very safe. And that, of course, leaves us to look at what are the processes in the various places. And at least compared to US, the European is complex, lengthy, and very expensive. And to a certain extent also unpredictable. On top of this, we also feel that it's not proportionate to risk of all the consumer safety, which I think could be something where we could work on becoming better. To my knowledge, at least no novel food have been approved in Europe or by EFSA in the recent years. And this means that the path to commercialization is very biased if you compare what we do in EU versus we do in US. And I'll come back to what happens in the US, where we have a pretty standard way. When the protein is known, the production strain is known as being safe, then it's still based on scientific proof that you can demonstrate safety. But if you can do that, then your way to commercialization is very, very fast. Months instead of years, and actually not very expensive. So at least I would highly recommend that we reboot the innovation for food in Europe, and that we may choose 3 to 5 bulk protein and fast track them and see what is doable. Because otherwise it's quite obvious that we will be years behind what goes on in the US. And if I have to put it into perspective, the BLD we have made in a fungal host was approved July 23rd this year in a grass self-affirmation by FDA. And all this we could do in around 6 months. We have not even put together our dossier to Europe at this point in time. And we'll still need to use another maybe 6 to 9 months just to put together, and then we start the process. So there we would at least propose that for some of these proteins which we have been consumed for hundreds, if not thousands of years, where there is safe technology, that we then at least find as a fast track to market, which I don't say it has to be the same as they do in US, but at least which mimics some of these things where we pretty fast can conclude that there's a limited amount of risk. The alternative is, if we don't do this, that then these proteins will take off in the rest of the world, and we will not then get access to much more environmental friendly production of a lot of the food we consume today. Again, the reason that I say this is that it's quite funny that because on the basic technology to deliver a lot of these proteins, Europe is in the lead. And therefore it is a little bit strange that the market which will take off will be all other places in Europe. And for me, from my point of view, we need to grow innovation in Europe. And of course, in my mind, in the long term, serve Europe out of European raw materials. And we can do that with this technology. So there's a lot of aspects in that we can take any protein, produce it microbially, and deliver it safely to market. Unfortunately, we are lagging a little bit behind on this, you could say, approval, and there has been good reason for where we ended as we did under EFSA. I'll just propose that we try to take a second look, and I've not seen what came out today. I will read that with big interest, but I think we should try to readdress that some of the bulk proteins which are really known as being, we have eaten them for a long time. How could we fast track these to market? And that's more or less input from my side. And then thank you for your attention.
Thank you, Per. That was a very interesting talk. Per has to leave after his talk, so it would be great if anyone has any questions to raise them now. But I have one question, Per. Obviously, you have prepared a self-affirmed GRAS, and you have affirmed as 21st Bio that your product is safe. In terms of R&D, how did you prepare for the regulatory requirements for preparing your self-affirmed GRAS, and also how are you planning for the EU novel food approval process?
Yeah, we made, of course, 3 individual batches of the protein. We did put the genetic construction together in a way so we know it can be approved in Europe, because, and that's quite funny, because in US, they don't know about this. So the US construction, they will never be approved in Europe, which is, yeah, you could say good or bad, but it can be put together in a way so it should be relatively straightforward to get it approved. Then the 3 batches, you have all the analytical documentation. Depending on what strain you use, there are certain potential toxins you have to make sure that they are not part of the genetic of that specific host. Fairly trivial standard. We have done that many times. But obviously, you put together this package on 3 individual batches, and then you go through a science board which validates what you put in, and that it's made by proper analytical standards, and that you also have produced your batches up to best standards. And when you are in that place, then it takes a month. Then FDA committee goes together, and then if there's no questions, you get it approved. So all this happened in 6 months. Obviously, we worked on the cloning and getting it right somewhat longer. It's taken roughly, in total, we are 2 years down the road now. Mm-hmm. And that means that the R&D part these days, we can do very fast, much faster than the regulatory approval. And therefore, of course, US get a huge advantage that you more or less get it approved as soon as you could put your data together, then it's approved. And I know that we have a different system in Europe, and it's not easy to change, because we have all these member states who has to agree, and it can go in national phase, and we have a parliament who also have to approve to this. But what we try to propose here is that, can we make, for something where we are very sure that this is safe, can we make a fast track to market? That's the proposal. And I know it's not going to be simple. I know that we will have most likely some of the farmers, we will have some of the local politicians on our back, but I honestly believe that we desperately need also to utilize, when we are on an innovative point of view, we are ahead of the other guys. It is a little bit annoying to see that they run with the markets.
Yeah, I think it's just important to clarify that the GRAS pathway doesn't constitute an approval. It's not a formal approval, so it's just to clarify that point.
Yeah, but it's generally recognized as safe, and of course, soon you get your number, then you can sell, or you can actually sell already from there has been no questions. Mm-hmm. Of course, it's still up. You have the full responsibility. If your product is not living up to the standards, it's you who have the responsibility. And of course, you have to have procedures to make sure that what you provide is safe.
Absolutely. And just one other point for clarification. I think in your slides, you said that no novel foods have been approved in recent years. And there have been many novel foods that have obtained approval since 1997 and since the new novel food regulation became enforced in 2018. But if you're talking about dairy proteins or proteins derived from precision fermentation, then yes, we haven't seen any approvals for dairy.
And I guess it also depends a little bit what is the definition of novel food. And I could push it a little bit more. If I'd made a few amino acid changes, which will make the beta-lactoglobulin more healthy with more branched amino acids, then it will not be something you could get from an animal. Then it will fall in the complete novel food. And I don't even think about what I then had to document. And it will be very, very easy for me to make something which was even more healthy, where the risk would be limited. So that's why I'm pushing this. And maybe it goes back to the definition of what is a novel food.
Well, actually, Karen has made a very interesting point. So she said, would it be a solution to reintroduce the substantial equivalence procedure in the EU? So this was a procedure that was permitted under the old novel food regulation, where if you're able to support substantial equivalence, then you didn't have to go through the whole sort of novel food approval procedure.
I think that would be a huge step forward. And actually, you could say the substantial equivalence, and I'm an old person, so I worked with that too in the old days. And most of those proteins we want to deliver, even though we make a few changes, then they are safe. It's actually all the other stuff you have to look after, not the protein as such. So I think it's and maybe not a lot of people understand this, but it's of course part of where we need to come together. And the substantial equivalence would be a great starting point.
So Claire has made a remark here. So if the EU does not start to accelerate approvals, what is the consequence for our own food security if other regions of the world have these? Can we have these? Can we do not? I'm also thinking of agriculture.
Yeah, again, we are talking, this is going to be a long journey, and I don't argue that we should get complete rid of the dairy products we have today because they taste good, they are relatively healthy, and we have been using them for a long, long time. But gradually, we need to make a change where we go to more sustainable production systems. And yes, this would have quite big impact also for the, you could say, supply security in Europe, that we could produce it locally. Maybe even down to that we have some of these factories, because they can be quite scalable. Even they can be on a farm site. So you produce them with local raw materials for the local market. So we get rid of that. We have to transport a lot of materials around the world, which long term, in my mind at least, doesn't make too much sense either.
Okay, thank you. And Yvonne Dommels has one last question. So did you test the allergenic potential of your protein?
No, because we know that beta-lactoglobulin is highly allergen, and it's used as an allergen in all allergenicity tests. And of course, these are inherent properties of proteins that they are potential allergens. So if you inhale them, or if you have a leaky gut, then there are of course some health associated risk by taking this. But it's not different from the dairy we eat today already. It's the product is essentially the same. So we know that BLD is highly allergen, and it's really used as a reference for allergenic response. So I think that's part of nature which we can't change. And in my former life, I maybe spent 15 years trying to make proteins less allergenic. And I would say we gave up because each time we took one epitope away, another one pops up. So it's a never-ending story. And until recently, I've only seen one protein, and that was a surprise for me that you could find a protein which are non-immunogenic. And surprise, surprise, that's spider silk. Spider silk you cannot raise antibodies against spider silk. And that was new to me. And it does actually give you a nightmare when you have to find it again, because then you don't have an allergenic tag you can bind to. but that's a separate scientific issue. But again, that goes deeply into the understanding of how proteins operate and behave.
Super, thank you, Per. I think we're ready to move on to our next speaker. So thank you very much.
You're welcome. Bye-bye.
Goodbye. Thank you. Very happy to introduce Muriel de Wilde from the bio-based Europe pilot plant. And Muriel is going to talk to us about the proof of concept to market. So how we scale and scale up and optimization.
Yeah, thanks for the nice introduction. Good afternoon, everyone, and thanks to the EuropaBio team for this very nice stage. So this is indeed our pilot facility in Ghent. Next, please. Yeah. And we indeed bring our customers from lab to fab, or from a lab-based protocol or basic protocol to an industrial protocol. Next, please. A bit of info on who we are. So BioBase Europe Pilot Plant was founded in 2008. We have approximately 57 fermenters ranging from 250 ml to 75,000 liters. So that gives a bit of a range of what we bring our customers to. We invested over 80 million euro in our pilot facility. We're not for profit because we received most of those investment budgets through public grants. We've grown to 170 employees in the past years. We're fully independent. We don't have any industrial shareholders. And we're a service provider. We provide scale-up services, and we also do that within European consortia. We're based in Ghent, in Belgium. And we've implemented quality management systems certified with the Food Safety FSSC 22000, an FDA registration. Next, please. So our mission is to enable the transition to a sustainable bio-based economy. And we do that through providing 3 types of services. That's process development, scale-up, and custom manufacturing. And all the products we make are bio-based. Next, please. Yeah. So our plant is a one-stop shop for the bioeconomy. We bring in biomass that can be first-generation sugar like glucose or sucrose, but also second-generation lignocellulosic sugars. Also side streams that need to be valorized, side streams from food industry, for example. And we also use gaseous C1 feedstocks in our gas fermentation projects. The technologies we have are biomass pretreatment, biocatalysis, fermentation, green chemistry, and we have a very wide set of purification equipment to make sure the final product meets the specifications. So we make food products, we also make novel food products, but we also make other products like cosmetics, chemicals, materials, preclinical therapeutics. Next, please. So some examples of novel foods that we produce or that we can produce that have been approved as a novel food, or that could be approved in the future, hopefully, or that some of the companies indeed chose to go for the GRAS status in the US and start the US market as their first market entry for those new products. So we make a lot of microbial alternative proteins, so egg or dairy proteins like lactoferrin, beta-lactoglobulin, casein, ovalbumin. So these products are of course produced because they can be produced in a much more sustainable way, in a safer way. And other examples are microbial sweet proteins like tomatin, brazine. We also produce single cell proteins, so fungal mycelia or single cell yeast products. And just on a side note, corn, so the Fusarium venenatum strain is not a novel food because it's been on the market since 1985. We also produce a lot of microalgae, and then to produce the biomass or specific products purified from them, like nutritional fatty acids or natural colorants. We produce human milk oligosaccharides and human milk proteins. An example, and in this example I can mention this is a customer of ours, in Bijos, a Ghent-based company that has a technology to produce a lot of different human milk oligosaccharides, of which several have been approved as novel food, for whom we produced the regulatory batches. An example of a human milk protein is human lactoferrin. Some dietary fibers that are extracted from second generation feedstocks. Microbial fats and oils are really coming up. And of course, specialty sugars, for example, arabinose that you can extract from sugar beet pulp, or kojibios that can be produced from sucrose via an enzymatic process. Next, please. But indeed, to build up a novel food dossier, quite some scientific data needs to be generated. I'm not a specialist, of course, but I just listed what needs to be measured or entered into the dossier. So a description of the novel food, a description of the production process, the compositional data, the stability of the product, specifications of the final product, nutritional info, toxicological info, allergenicity. And next, please. And all this has to be analyzed on at least 5 representative batches produced independently, produced at an industrial production scale. The process description has to be detailed enough to give info on safety and nutritional value. The operational limits and key parameters of the process have to be described. There has to be a production flow chart. And just a separate comment, if you want to produce a non-GMO product, all recombinant DNA needs to be removed down to the level of detection and no viable cells present. Otherwise, you have to comply with the GMO legislation. Next, please. So how do companies do all this? Because we all know the valley of death. That's, of course, the place where you want to translate a lab-based protocol to an industrial protocol. That means you have to have industrial equipment. You have to run the process in that industrial equipment that is very CapEx intensive. It's also very personnel intensive, so OpEx intensive. But you just don't have that money yet if your product is not on the market. So that is exactly where the Biobase EuroPilot plant steps in. We have all this equipment. We have skilled and experienced personnel. So our customers pay us a service fee just for the time and the scale and the operations we perform for them. So next, please. So we make sure that the equipment is industrial equipment. It's state-of-the-art. We have maintenance plans, validated cleaning procedures. We have the quality certifications, experienced personnel analytics to make sure we comply with the final product specifications before you really have to build your own plant that will otherwise be very uncertain. So we can produce your regulatory batches. We provide the full report with all the equipment used, the settings applied, the process data and mass balances generated, the analytical data, and the process flow diagram. Because this valley of death is already a very, very big challenge for companies implementing new products. And as you see from this estimate of novel food application timelines. You're on for at least 30 months if there are no questions about your dossier. So it's very important to make a good dossier. So we're for sure ready to help companies do that. Next, please. So typically when a customer comes to us, they have a basic protocol that we can further develop if needed at bench scale to make sure the proof of concept is done. Then we go to scale up. There you can already have a sense of the economic viability of your process, the robustness, efficiency, and scalability, and you can demonstrate in industrial equipment what the process will look like at industrial scale later on. You can test the final product quality specs and adjust the process if needed to achieve them. And once we've optimized and validated the process, we can do custom manufacturing at the largest scale we have available. So our customers can enter the market before they build their own plant and raise additional financing rounds. Next, please. So which equipment do we bring in exactly? In terms of fermentation, our lab fermenters range from 150 liters over 1,500, 7,500, 15,000, and 75,000. We sometimes get the question, at what scale do these regulatory batches need to be run? There's no straight answer to that. That really depends on the entire process, how representative it is for an industrial scale. For some processes, that can be the 150 liter scale. For other processes, you would have to go to 1,500. And of course, if you want to meet other objectives of a scale-up, like generating product for application testing, you might want to go to larger scale, 15,000, to have more product available for that. In the DSP, we have this very wide set of unit operations. Maybe if you click once more, the title appears. So cell lysis equipment, high-speed separators, filtration, like cross-flow filtration units, ion exchange, chromatography, solvent extraction, crystallization, and drying at all those different scales, because of course the purification equipment needs to match the fermentation scale. Next, please. But another very important aspect, besides production of regulatory batches and optimization of the process, is partnering up, making sure you meet the right companies that can serve you and help you in all the other aspects like financing, developing business models or life cycle assessments. And that's why we also organize an event. It's in December in the airport of Brussels. So we will have 100 startups pitching their technology to investors. We will also build an ecosystem around open access pilot facilities. It's called Pilots for You, and it will be extended with the co-pilot project. Next, please. Because bringing an innovative product to the market is really all about teamwork. So this is our team. And the next slide, please. Yeah, that's my presentation.
Super, thank you so much, Muriel. We'll defer and wait until the end of all of the other talks to have a Q&A session. But if you have any questions for Muriel, please type them in the chat and we can ask her at the end of the session during our Q&A. So next up, we have a very exciting talk. I don't know if anyone else has been up since 6 o'clock this morning waiting for the updated novel food guidance to be published, but I certainly was. So today has been a very exciting morning already. So today we have Umalaus Viveris, who is the scientific officer, nutrition and in the Nutrition and Innovation Unit in EFSA. And so today, Omalaus is going to be talking about the novel food risk assessment and also give us insights into the updated scientific guidance.
Thank you a lot, Hannah, for introducing me, and thank you very much for having me here in this webinar. So indeed, today we're going to talk about the updated scientific guidance that was actually published just a few hours ago. In the next slide, just some brief information about like my background. I have a background in chemistry and then some master studies in food chemistry technology and animal-derived foods. And then PhD studies on risk-benefit assessment, particularly assessing what is the health impact of substituting red meat with alternative protein sources. I have some work experience in food R&D and food safety sector, and since 2016, I'm working at the European Food Safety Authority at the Nutrition and Food Innovation Unit, and more specifically on novel foods. So in the next slide, just a brief overview of what is EFSA. I assume that most of you know already, but for those who don't know, EFSA was established under the EU law in 2002, following a series of food crises in Europe. And the aim, EFSA was part of a project aiming to improve the EU food safety system, help ensure a high level of consumer protection, restore and maintain the confidence of consumers in the EU food supply. And this project also aimed to clearly separate the risk assessment and the risk management functions in the European Union. In the next slide, we have already heard earlier what is a novel food in the European Union. And just considering what examples you have seen already, you can understand that novel foods are very heterogeneous. It's a very heterogeneous field. We can have products that are single substances, but we can go up to very complex mixtures or even whole foods. In the next slide, you can see the briefly the authorization procedure in the EU to place a novel food in the market. So first of all, please have in mind that the dossier is submitted to the European Commission according to the novel food or the traditional food guidance that have been issued by EFSA. Then the Commission, in collaboration with EFSA, have to assess the validity of the dossier to ensure that the minimum information requested to start the assessment is there. And then when everything is ready, the Commission sends the mandate to EFSA to start carrying out the risk assessment for novel foods. For traditional foods, we don't carry out the risk assessment, but in collaboration with member states, we raise any safety objections we may have. So to do the assessment for novel foods specifically, we have by law 9 months. But of course, if something is missing from the dossier, or if some of the elements of the dossier trigger additional data requests, then the time will stop and we can go back to the applicant to request additional information in order to investigate the safety of the product. As soon as the outcome of EFSA is, the scientific opinion is ready, it is published and is sent to the European Commission and the member states, who are the risk managers in the EU, and they decide if and how this product is going to be placed on the market, of course, considering the scientific advice that has been provided by EFSA. And all the novel foods that have been authorized in the EU are included in the Union list of authorized novel foods that is available online. And then someone can see what has been authorized, which are the specifications of this product, and any other specific requirements linked to these products. So in the next slide, briefly, what we do not do in the area of novel foods is to develop food safety policies and legislation. We don't adopt regulations, we don't authorize marketing of new products, or it's not us, EFSA, defining the labeling, and also we do not enforce the food safety legislation. What we do is to provide independent scientific advice and support to the EU risk managers and policymakers on food and feed safety. We develop and provide up-to-date guidance in the field, and also we communicate independently and timely what we find during our risk assessments. And of course, last important thing that EFSA does in this remit is to promote scientific cooperation. So now we are going to focus today on the second part, developing and providing updated guidance. So we received the mandate from the European Commission back in June of 2023 with an adoption deadline in June of 2024. And the risk managers asked EFSA to do this update because there were some regulatory updates to be considered in the field of novel foods in Europe. Also, risk managers requested EFSA to reflect our experience in assessing novel foods and also to capture any new advances in science and technology in this field. And when I talk about EFSA's experience in novel foods, is due to the fact that since 2018, the assessment of novel foods has been centralized in the European Union, and EFSA receives all novel food dossiers, and you can imagine that we have many of those and many different novel foods. There are many also EFSA issued cross-cutting guidance documents that are applicable also in the novel food field. Also, we have advanced some of our risk assessment methodologies. EFSA has also developed some new tools that can help both risk assessors but also applicants in the field. And also we have received feedback regarding the novel food guidance through different engagement activities, like for example, the colloquium we had last year in Brussels on cell culture derived foods and food ingredients. In the next slide, you can see, as I told you today, that the guidance was published today, a few hours ago, and it's applicable to all novel food applications that are submitted to the European Commission from 1st of February of next year onwards. And this guidance, just to highlight that it's about the scientific aspects, but in parallel, in a few days, EFSA is going to issue the administrative guidance for the preparation of novel food applications in the field of novel food. And this is the other document that helps with some administrative and practical aspects with regard to the submission of novel food applications. On the next slide, you can see again the contents of the novel food guidance. Let's say that the main sections have remained the same. We did some reshuffling towards the end of the guidance. And again, we talk about information on identity of the novel food, production process, composition, specifications, history of use, proposed use and use levels, ADME data, toxicological information, nutritional information, and of course, allergenicity. In the next slides, we're going to see in the different areas of the novel food guidance some of the main changes, of the main updates, and of course later with the questions we can also dig into further in anything more specific you would wish to. So in the area of identity of the novel food, we have expanded like the information on how the applicants can consider which is their novel food. Because from previous applications we have received, we have seen that many times applicants have difficulties to define which is their final product. And in some cases, we receive data on formulations that they include materials that are not novel, on formulations that are preliminary products. So we try to explain this concept a bit in more detail. Also, we give some more insights when novel ingredients can be considered part of the novel food. And also we provided some information on how the applicants can better name their novel foods, because the name, of course, is a very important descriptor of these products. In the different sections, we have again a section about chemical substances and products of mineral origin and polymers. We kept the section about novel foods from animals or their parts. We have enriched the section about products consisting of, isolated from, or produced with microorganisms. And also we have provided more information about products that are derived from cell culture or tissue culture, from animals, plants, macroscopic fungi, or macroalgae. And also, we have revamped a bit the information in the area of plants, macroscopic fungi, and macroalgae. And we provide some more clarifications for novel foods that are consisting of or they contain engineered nanomaterials. On the next slide, you can see some more information about these categories. For the chemical substances, polymers, and You can see that we have provided some more suggestions for new identifiers to be provided, some further insights into identification methods and characterization techniques, also how the applicants can consider finding suitable comparators, and also we can include some reference to ECHA guidance about identification and naming of these products. Regarding microorganisms, novel foods produced containing or isolated from microorganisms, we have expanded the information on the role of these microorganisms in the novel food production. We have provided more information about the cases where the novel food is produced using gene production strains, which is the purpose, characterization, and structure of genetic modification should be explained. Also, some further information about viable cells and DNA of production strains, and also some more information on the QPS status of microorganisms, genes of concern that have to be investigated, and when whole genome sequencing data are needed. For the plants, some information on the experimental verification of the identity of the plant versus the growing regions and how this can be affected, and also considering the harvesting season. And also, we require now a non-GMO statement for plants, for the plant source. And also in the next slide, you can see that this non-GMO statement is asked also for the animal-derived novel foods, for which again we asked verification for identity of the source, the suitability of the source for human consumption according to EU applicable frameworks, and also we request for more information on animals' health status and the origin of the initial livestock. Then we move to the cell culture or tissue culture derived foods. We have added more requirements for established cell lines or primary cells, and the request for compliance with inspection requirements, and also the absence of zoonotic agents. And also, what is requested new is information on whether the cells or tissue sourced from a non-GM source have been genetically modified to produce the novel food, because then different regulatory frameworks may be applicable. And last, in the engineered nanomaterials, we have cited the new EFSA cross-cutting guidance documents about the risk assessment of nanomaterials in the food and feed chain. And also, it was highlighted that these engineered nanomaterials is something different from small particles, novel foods including small particles, including nanoparticles. And this is something also that is stressed in the composition. In the next slide, you can see some new aspects linked to the production process of the novel foods. There is a paragraph talking about general provisions, how to provide information on the input material, materials compliance, the production yield, novel aspects of the process, quality and safety assurance parameters, and also standardization criteria for the novel food. There are also considerations for specific production process steps. For example, how to describe the conditions and the farming practices, the culture conditions, when also we tackle cases when food enzymes are used, and what happens when this food enzyme is already assessed by EFSA, and what happens when this enzyme has not been assessed yet by EFSA. And also we have added another subsection about consideration for specific novel food categories, about plant, fungi, algae, or animal-derived novel foods, chemical synthesis-derived novel foods for which microorganisms are employed in the production process, and also novel foods that are cell culture or tissue culture derived. And there are also some additional considerations to help the applicants, especially in cases when the novel food can be produced with different production processes, or in cases in which the novel food application contains data that derive from different food producers, for example, in case of a consortium. Then we also there describe specific consideration for these cases. On the next slide, about composition, we have explained a bit further the role of this kind of data in the risk assessment, and then different subsections, of course, the general requirements, but then more specific information on the analytical methods to be employed, how applicants can address compositional variability, some information on sampling practices, and of course, compositional analytes, how the applicant can decide which substances, which components are to be analyzed in the novel food. And of course, there are more specific requirements differentiating between single substance and simple mixtures, and also novel foods that are complex mixtures or whole foods. And last but not least, under compositional data, the stability of the novel food. And here we have created a subsection about the impact of processing of the novel food in the proposed for use matrices. Of course, these are the high level information. There are more details and more changes. But for example, now the minimum number of batches is well defined in the guidance. So we can also go deeper later if you want with regard to any of these topics. On the next slide, about specifications, again, we try to explain more the role in risk managers. We ask for the inclusion of key descriptors, source names, microbial strains, production process details, major constituents. The applicants have to justify why they chose something and why they propose specific limits for this specification parameter. And also a reminder that specifications should be verifiable using the indicated analytical techniques with information, of course, on the method sensitivity alongside the proposed specification limits. On the next slide, it's about the history of use of the novel food and its source. Not many changes on this slide, but here again, we request information on the use of the novel food as food in countries outside of the EU, information on non-food uses, the extent of use, population groups for which it has been a dietary component, the role in a diet, and also any relevant information on handling and preparation techniques. But we also, the applicants can also provide information on the history of use of the source, because not all novel foods has history of use elsewhere in the world. And again, more or less the same way of thinking is applied. On the next slide, you can see some changes that we have done in the area of proposed uses and use levels and anticipated intake of the novel food. According to the new regulatory changes, novel foods cannot be restricted to a group when added to whole foods as ingredient. So in this case, the target population is the general population, and this is specified, but it's also specified when an applicant can restrict the target population to specific subgroups. We have included requirements for food ingredients, whole foods, food supplements, and particular food categories with regard to proposed use and use levels, and also some new tools like the Diatex tool, but also some older tools like the FAME tool, in order to calculate the anticipated intake of the novel food, in order to use information for the exposure assessment. And also there are considerations regarding the combined intake, considering other sources of the novel food or its many constituents, and also information about exposure to substances of possible concern. On the next slide, you can see this graph. It combines ADME information, toxicity testing information, including genotoxicity and repeated dose toxicity. And this is a tiered approach, and you can see the different tier requirements for each one of these three, let's say, subsectors, ADME, genotox, and repeated dose toxicity. And you can see what is requested in each tier and how findings from a specific tier can lead to more scientific required evidence in the same sector, or in some cases, how findings in one tier can lead to more to the need for more evidence across different sectors. For example, how findings in tier 3 of ADME can lead to the request for studies in genotoxicity and repeated dose toxicity. Or, for example, you can see how positive or ambiguous findings suggesting potential genotoxicity can trigger requests for other studies in repeated dose toxicity, investigating mechanistic aspects of and mode of actions. So this is the new tiered ADME toxicity testing approach. And actually, some of the parts of this approach have been updating, considering also the feedback that we received during the public consultation of the guidance. And the same, of course, applies to the other areas of this guidance. On the next slide, you can see some information about nutritional aspects. We explained more the concept of nutritional disadvantages in the novel food risk assessment, and there are considerations regarding the excess intake of nutrients, inadequate intake of essential nutrients due to the novel food consumption, specific consideration for novel foods proposed as a new source of macronutrients. And here it's worth to highlight that today there is also another guidance on specific principles and data requirements for the safety and relative bioavailability assessment of new macronutrient sources published. And it's a document that should be read in conjunction with the novel food guidance when, of course, this is needed. And there are also specific considerations for novel protein sources as novel foods, and also some additional considerations, for example, when in vitro in silico animal models can be used, and how to deal with data from human studies in order to investigate, for example, the interaction of novel food with the diet and other nutrients. On the next slide, you can see a flowchart. It's that guides applicants how to investigate the nutritional impact of the novel food, of course, under the proposed conditions of use. So you can see here considerations that apply to all novel foods, and you can see the different steps. And also you can see here also considerations that apply to novel foods that intend to replace other foods that are already in our diet. So you can see that there are different, let's say, outcomes in this flowchart, and you can see that there is also some proposals in cases that an applicant, while preparing the novel food application, finds that the consumption of the novel food could be nutritionally disadvantageous. There are some proposals how to tackle these preliminary, let's say, findings. On the next slide, which is the last section of the guidance, allergenicity, you can see it's, I would say that it's one of the most revamped sections of this guidance. You can see this flowchart about considering different production processes of the novel foods, different categories of the novel foods, and different, then all this leading to different data requirements. And it's also another tool that the applicant should consider when investigating the allergenicity potential of the novel food. And you can see the different categories. We talk about novel foods with no protein derived from the production process, novel foods derived from allergenic foods subject to mandatory allergen labeling, novel foods derived from allergenic foods that are not subject to allergenic labeling, and then also novel foods for which the allergenic potential is unknown. And on the next slide, there is another tiered approach to be used by the applicants about novel foods for which the allergenic potential is unknown, with specific mention to single proteins and simple protein mixtures. And you can see here a tiered approach again, how the applicants should consider collecting evidence, assessing this evidence in order to pass, if needed, from tier 1 to tier 2, etc. And when discussing these elements with the member states, EFSA tried in the guidance to put some requirements that are both informative, but also they don't go really beyond what is really needed for the risk assessment and later to be considered from the risk managers. So this was my last slide about the updated sections of the guidance. In the next slide, you can see that there is still a call open for support to novel food, small and medium-sized enterprises. And of course, this call ensures confidentiality of information provided by the participants. And this aims to provide advice. EFSA provides advice to SMEs in a telemeeting via the General Pre-Submission Advice Service. And we can have early briefing meetings for small, medium enterprises still working in the development of the novel food. But also we can have pre-submission meetings for SMEs that are almost ready to submit their application in the system. And this call is open until the 31st of October. So it's like 1 more month open. And I have put here on the slide links that you can use to go to the call page, to attend the pre-recorded info session that EFSA had delivered. And also, if you have any doubts, you can reach out via the Ask a Question function of EFSA. And in the next slide, I would like to inform you of a conference that EFSA organized with other entities like BFR, FDA, the Singapore Food Agency Authority, and also MATIS. It's about the International Conference on Alternative Proteins for Food and Feed. It takes place in Berlin, beginning of December of this year. Also here I have included some information for you. And if you want to know more about EFSA's work, etc., on the next slide, the last one, I have included the different channels through which you can stay connected with our work. Thank you a lot for your attention.
Super, thank you so much, Emalaus. That was incredibly informative. Lots for us to digest about the new guidance. I just wanted to make a quick remark about what the next phase is after the EFSA risk assessment. So after EFSA has published their opinion, then the risk management phase starts. And here the European Commission prepares a draft implementing regulation, which is reviewed by the Standing Committee of Plants, Animals, Food and Feed, along with the EFSA scientific opinion. And member state representatives vote by qualified majority voting. And to obtain approval, 55% of member states representing 65% of the EU population must vote in favour. And once approved, as Emalaus already mentioned, the novel food will be listed in the Union list of novel foods. So moving on, we're now going to hear from Timon van Haster, who is going to talk to us about how cultivated meat got to Dutch plates. So Timon is the Public Affairs and Communications Director from EuropaBio. So over to you, Timon.
Yeah, thank you, Hannah, for the introduction, and also EuropaBio for hosting this webinar. So I'm going to talk you through the way we brought cultured meat to Dutch plates. But first, a bit about me, a quick introduction. As Hannah said, I'm the Public Affairs and Communications Director at EuropaBio. I've worked there now for about 7 years. Background in strategic communications, public affairs, stakeholder management. That's about it. Maybe some more about EuropaBio itself as an industry association. What we aim for, what we work for is the biotech makes life better. That's our motto, so to say. And if you look at our membership, if you go through the next slide and the next one, I think we represent about 280 member companies, all working on transformative innovations across the whole biotech spectrum. So diagnostics, medicines, vaccines, but also cultured meat, proteins, materials, crop seed breeding, etc. I think what distinguishes our membership is that about 85% of them are startups, scale-ups, and SMEs, and the rest of them are established or large companies. So what do these companies have in common? Well, they're actually quite different, but they share a lot of the same challenges. So they're all very knowledge intensive. They all need a lot of funding, really capital intensive. And whether you work on medicines or cultured meat or new crops, you have a long way to market. It's complex, it's highly regulated. So those are some of the shared challenges. Maybe going on to what we will be going into today is really the road to the tasting sessions. So how did we manage to make these tasting sessions of cultured meat and fish in the Netherlands possible before filing a novel foods dossier? Well, if we look at the process, well, like you can see immediately, it's taken a long while. It's taken about 6 years from the start until the actual tasting sessions themselves. And the way we worked on this is that we came across, we have a large number of frontrunners in the Netherlands when it comes to cultured meat. A lot of the technology has also been invented in the Netherlands. So we have a couple of companies that are leading the way, and they came into the issue of, well, we're developing these products. We have a long way to go. Are there any possibilities to help going this sector going forward? And one of the possibilities that we discussed is, well, maybe it would be good and useful and interesting to have the possibility to have these tasting sessions in a safe and in a controlled way that allows companies to actually, well, experiment with the taste and the texture, to take the general audience a bit along with this innovation. So that's something we started working on in first instance with some members of parliament in the Netherlands. And well, it took a while, but well, midway 2018, we found a member of parliament who was really enthusiastic about this sector, about his innovations, and he posed the first questions on cultured meat and the possibilities for tastings. So it took a while for our government to get back to that. We had a couple of instances in our parliament where there were hearings and questions. In the end, our then minister Schouten came back to parliament and said, well, it's all nice and easy, but cultured meat needs to go through the novel foods procedure first. So there was a first well, step back from our point of view, but not something we let it down with. So we started again in February 2019. We then had two members of parliament who were very supportive of this topic. They actually filed two motions to really call for the government to support cultured meat and the sector and giving them more space to work with on their products. That actually took a while for the Dutch government to get back to. There was the first cautious confirmation in June 2020 that tasting sessions could be possible before filing for novel foods applications. But that was right before we had elections in the Netherlands. This government really did not take action any further. So we had elections in 2021, and before a new government and new members of parliament were active, who were actually in February 2022, before one member of parliament we still knew and one new one that actually went for it and put forward a motion that was adopted quite broadly on really the request to our government to explicitly make possible tasting sessions in the Netherlands in a safe and in a controlled way, but before filing for a novel foods dossier. But that's the period that we got together also with the Dutch government, so with the Ministry of Health, with the Ministry of Agriculture, with the companies working on cultured meat and fish, and with some experts, to really develop a code of practice that was well, a guide for hosting and organizing and assessing these tasting sessions. And that took a while, but in the end we had in July 2023 the code of practice ready. And then after that, it took again, well, a half a year, a bit more to actually come to a tasting session, because we needed to set up a committee of experts that were assessing the applications by companies. A procedure needed to be set up, and it's now hosted by the Cellular Agriculture Netherlands Society, which is a public-private entity funded by the Dutch Growth Fund. So in the end, in 2024, we had the first tasting sessions. And if we go on to the next slide, the first one was in April 2024. It got a lot of media attention. It was hosted by Meatable, one of the two major Dutch frontrunners in this field, together with Prins Constantijn, Ira van Elen, the daughter of the famous inventor of cultured meat, and with a chef who's very famous, has Michelin stars, and a host of other journalists and media that also were allowed to taste these products. This is the first official tasting session in the European Union. This was the second one, took place at Mosameat in July 2024, also again with a diverse panel of stakeholders and audiences. So that leaves us with the question, well, what can we learn from these tasting sessions and the process that led up to that point? So if we move on, what we see looking back at this whole process is the backing of a number of key ambassadors in our parliament. In the years that it took to get this off the ground, it really was crucial. We worked with members of parliament that were either in the government coalition or close to it, and that were able to generate support among several parties, that were actually seeing this as a topic they really cared about, that they really wanted to move forward. What we did in a broader sense for members of parliament is, aside from conversations, we did working visits. to these companies themselves, to ecosystems that work on cultured meat. We also hosted some hearings. We send them a lot of examples. I don't know if you're familiar with it, but a couple of years ago, Gourmet also had a sort of a tasting in Paris with their foie gras that was documented by Bloomberg, and that really helped, well, visualizing what a tasting session could look like in a good and safe and controlled manner. And that really drew members of parliament in. What we saw in first instance is that policymakers did not really want to touch upon tasting sessions and making it possible in the Netherlands. So they pushed it back quite a lot in the early phase. They delayed it a bit, but once this motion was actually there to make this possible, and it gained a majority in the House of Parliament, they actually caved in and really worked together with us as companies, as sector representatives, and other experts to really get this code of practice, a sector-initiated way of determining whether tasting sessions can be held in a safe and responsible manner to make this a reality. And we're actually quite proud that we managed to do this in co-creation with companies and ministries and regulatory experts. And we also see that there was really a broad support for certain conditions that were built into the code of practice. So having this assessment by an expert committee with several disciplines in there, toxicology, for example. There's also a condition that companies that want to organize tasting sessions need to have a base of activities in the Netherlands. It's on one hand a limiting factor, of course, because we got a lot of questions from companies all over the globe whether they would be able and allowed to have tasting sessions in the Netherlands. This is really to manage the work of the assessment committee. So that was something we agreed upon in the end, and also the use of GMO in the whole process, because it's not placing a product on the market. It's allowed for in these tasting sessions and in the code of practice. That's also something we are really happy with, that we managed to get that off the ground. So that's it about the process. If you look at the outcomes of these tasting sessions, first and foremost, the goal was always to support the product development of the cultured meat and fish companies that we have in the Netherlands. So to allow them to have these tasting sessions for customers, for key opinion leaders, to really move their product development forward. Additional targets or outcomes is also, it really helps to involve the current investors, to really show them what this is what we're working on, to actually give them a taste of what they're investing in, and also to attract potential new investors. Another one we think is also very, very important is that it raises awareness of cultured meat and fish with the general audience, because, and it's also something you see broader in Europe, there are some dynamics in which you see that cultured meat is labeled as lab meat or other negative connotations with this type of food. But what you see in these tasting sessions, you also saw it in the pictures just now. It's really a very ordinary type of food. It actually tastes like ordinary meat. So that's the good thing about it. And that's something we want to make the general audience more familiar with. If you look at the media attention it generated both in the Netherlands and abroad, with the pictures and stories, most of the comments by the tasters, whether they are journalists or chefs or other stakeholders, is that these products are actually quite normal and meat-like, and that's actually the biggest plus of these sessions. Well, more outcomes. It of course helps the position of the Netherlands as a biotech frontrunner. That's also part of the ambitions we have from Holland Bio to establish the Netherlands and that image. And that it also generates a lot of interest and questions from companies abroad. And we also think and hope it may serve as a base for discussions about optimizing novel foods procedure in the EU. And of course, there's now a renewed guidance, and we hope that it's a big step forward anyways. But also, as previous speakers have mentioned, we'd like to optimize and make it as fast and responsible as possible in the EU compared to other geographies. So maybe some of the next steps we see. The Code of Practice was initially established as a pilot phase that runs for a year. So that will actually run out in the first quarter of 2025. And pending the evaluation, there are a range of possible outcomes. One of the outcomes could be that this pilot will be terminated. We do not see that as a very likely outcome, but it still is one of the possible outcomes. Another possibility is that this pilot will be continued for a year and then evaluated again. And it could also be that after this 1 year phase, this could be part of the standing policy in the Netherlands. So these tasting sessions will be possible on a permanent basis. What we will also keep on doing is, well, work on the novel foods or the pre-novel foods tastings to make them also possible for other products that need to run through the novel foods, like products from innovative fermentation. We hope to establish those as soon after the evaluation of the cultured meat sessions. And we also will be working with members of parliament, for example, in a debate that is coming up in Dutch Parliament next week on biotech. And from a more broader sense, we also aim to contribute to improving the novel foods procedure, like I mentioned, because there's still a lot of hurdles left to tackle in this procedure compared to other geographies, and we need to optimize this as much as possible. So that's it in a nutshell, and I would like to thank you.
Super. Thank you so much, Timon. That was a very informative talk. Really great that the Netherlands has this tasting protocol and framework in place. I really think it's an excellent example for other member states to follow. So next up, we have Anders Nelving, who is going to talk about the consumer perspective. So Anders is the line solution manager from Tetra Pak. So Anders, over to you.
Thank you, Hannah. Pleasure being here today. Thank you so much to EuropaBio for the possibility to speak here today. So my name is Anders Nelving. I'm representing Tetra Pak. I work as a line solution manager, basically meaning that I handle our portfolio within the area of new food, but I also work in sales. So next slide, please. Tetra Pak, we are a privately held company. We are based out of Sweden and Switzerland. We are active in more than 160 countries around the world. We're soon 70 years old, and we're privately owned. Most of you maybe know us more as a packaging company. We are also highly involved with 5,000 employees in the processing area, bringing processing equipment to the market, and also servicing the equipment that we have installed. In all, we are 25 colleagues serving the world out of 80 offices around the world. Next slide, please. We are active in 8 categories. It all started back in the 50s with dairy applications. These days, we've added ice cream, beverage, prepared food, cheese, powder, plant-based, and now lately new food. Curiosity, in ice cream, the likelihood is that you will eat an ice cream, which is, it's high, more than 50% likelihood that the ice cream you eat has been produced in a Tetra Pak machine than that it hasn't. So we're much more than a packaging company, even if our packages are the ones you find on your breakfast and dinner table on a daily basis. With our size also comes a number of different services that we offer our customers and offer to ourselves. And one of them is consumer insights. And that is what my talk will be all about today. We use it to develop our portfolio, and we hope that we can help our customers and also the general public with easy to understand information shared in a nice way. So next slide, please. This first slide is really all about the headline. It says, Consumer Trends 2022. And many believe personal food choices can contribute to a better world. This, if we go back into 2021, 2022, this was when, you know, this generation of new food really kicked off and coming to everybody's attention. And the main message being the consumer is active and the consumer is wise. Now, next slide, please. On a biannual basis, in Tetra Pak, we do a study, or we compile a report that we call Trendipedia. The 2025 Trendipedia is soon coming. The slides that I will be presenting here today are basically some of that material, so soon to be published. And if we look at the consumer trends and the category trends that we see globally for 2025, they are as seen in yellow. We call it the savvy and the conscious consumer, and that translates to a category value of or category trend as pursuit of value. We have the mindful and the wellness category, or the trend with the consumer, transforming into a category trend of what we call a wellness wave. We have the green category, which is the sustainability part, both from the consumer trend and the category trend. And if you look now on the right-hand side, you will see that these three are the most prominent trends. On the x-axis, you see that they are high in prominence. They have a high impact. And if you look on the y-axis, the further to the north of the y-axis, the longer the trend is anticipated to withstand. So the red, purple, and the blue category, they are still there, but they're not as prominent and they're not as long-lasting if you look on a global level. When we take into account then what is a new food and are these trends relevant for a new food, our understanding when speaking to customers and speaking to the end consumers is that yes, our consumers are very conscious about the green aspect, especially in Europe. We see clear regional differences, but you will not sell a product just based on it being sustainable. It needs to taste good, it needs to have good benefits for me, and it needs to be reasonably cost comparative to the corresponding products on the market. Next slide, please. Trends develop over time, and this is now just an example on the health and wellness trend. So it's been around for 10 years. It's been around in these Tetra Pak Index and Tetra Pak Trendipedia reports for approximately 10 years. But it has changed a little bit over time. In the beginning, it was very much good for me. Whereas today, as you see in the bottom of the slide here, it's very much good for me and good for the planet. It's also changed slightly from being a very conscious consumer to being a very aware and maybe to some extent requesting consumer. In the beginning, the consumer took the portfolio or took the number of products that were there and combined it into something which was good for them. Today, they are actively seeking great products that can help them with health benefits and sustainability benefits. Next slide, please. Of course, there are regional trends. As mentioned earlier, the green trend is quite predominant in Europe. The reason why it's not seen as the highest influencer is that in other parts of the world, it is not as prominent. But for new food, we also have the aspect of significantly different regional regulatory framework. Not all products are available everywhere in the world. We've heard previous speakers mentioning the fact that Europe is very, we are far ahead when it comes to innovation, but we don't have the market to put our products. In a paper earlier this spring, we in Tetra Pak, we wrote, differences in food safety and regulatory standards are usually a combination of traditional, cultural, and political factors. Now, if you remove the political factors, to some extent, trends are very similar to you know what also runs into a regulatory framework. They're also a combination of traditional and cultural aspects. Why does one trend become a mega trend in one area and only a reasonable trend in another? On the right-hand side, in the bottom, is a questionnaire that we put out to, I believe it was 5,000 consumers in different regions in the Tetra Pak Index report of last year. The Tetra Pak Index report is available for everybody. You log on to the Tetra Pak website, and it's a consumer trend report compiled every year for public information. It builds very much on the Trendypedia results that are more of an internal document. But as you can see in the bottom hand corner, number one, the top orange boxes, basically the question is, have you heard about, what is your status on microbial proteins? And 58% say they've heard about it, but only 8% say that they have tried it. Now, the good part is that when you look into each and individual aspect down below, it says healthy versus unhealthy, it says nutritionist versus non-nutritionist. Most of the time, there is a strong favor for the positive aspect of the novel food, or in this case, the microbial protein. But you also see that there are small flags to the left and to the right of these bars. If there is a flag, that means that that specific region is significantly differing from the average index. So for instance, and maybe you find it surprising, maybe it's due for different reasons, but you can see, for instance, that in Germany, there is a stronger preference than the index, saying that this is bad for the planet. Whereas in other parts of Spain, for instance, there is there's a belief that it's used by many people rather than fewer people. So trends are different, trends change over time, but the longer term perspective, there are bigger trends that persist. For instance, now we see both the green trend, the health trend, and the price parity, the money value for money trend. Those will be with us for a significant period of time moving ahead, but they will probably differ slightly with the focus every year. Next slide, please. So in Tetra Pak, we do believe that new food is the future, but new food will come to the consumer's appreciation by tasty, healthy, and innovative products. You will not sell a product based on claims of the environment. The environmental claims, we believe that in a very soon future, they will be a must-have. They will not be a key driver. They will basically be a need for any kind of product. We see that the tasty products come through innovation. They come through collaboration. We see that the value for money can come from tying into the existing infrastructure. Currently, many of the products are that are coming to the market, they come with unique claims, they come with a unique process, and nothing is really standardized. And that brings an additional lengthiness to the approval process, but it also comes with an extra price. Moving ahead to bring these tasty, healthy, and innovative products to the market in a quick and cost-efficient way, we believe that the more you can tie into the existing food chain, the better it will be. Next slide, please. And we see these products, they are coming to the market. Especially we see them prominent in the plant-based area, where the possibility of adding a novel ingredient has the possibility to either boost the nutrition, or it has the possibility to replace an ingredient that has a bad taste from previously. So the plant-based segment is very prone to bring in novel food ingredients. But we also see it coming in the dairy industry. Here it's perhaps not as much taste related, but rather bringing the environmental aspect by being able to replace certain components in a better way. Possibly you can find a better carbon footprint or environmental footprint. So we definitely see these hybrid products coming to the market. There are, of course, as Timon, for instance, was mentioning, there are the products that are, fully new food, cultivated meat, for instance. Tetra Pak is not for the time being fully into cultivated meat. So I don't have any data to share specifically on where the consumers stand on that segment of new food. Next slide, please. Bringing innovative products to the market, it is a quite lengthy process. We have a history as a company to help our customers to drive this process, and we believe that we also have an established process to do this moving ahead. What we specifically would like to emphasize on is the box here on the bottom of the graph, where it says existing production infrastructure. Talked about it already, the definitions and communication, and also the food service sector. Often we refer back to the consumer as the individual consumer. We do believe, and we do think we see in the political segment, that the food service sector and the business-to-business sector is a consumer that should not be neglected by the startups and the new novel food industry. They will, in our opinion, have a huge impact on bringing this to the consumer. What we eat in restaurants, what we eat in airplanes, what we eat when we don't cook at home, has a high potential of being the door opener to this new segment. When it comes to the definitions and communication, we fully agree with what I think it was Timon saying it in the last talk. What is it lab-grown meat? Is it cultivated meat? Is it micro-fermentation? Is it precision fermentation? The vocabulary, to some extent, it's not always aimed at the consumer. Sometimes it's more aimed, for the time being in Europe, at the regulatory agencies. Here, we do believe that we together can improve and make it more both visual and easy to understand. Next slide, please. And here we do fall back to what other speakers have said previously. This is not something that one company or one individual does by his or her self. This is a combined effort. We have the makers, we have the producers, we have the startups, we have the governments, and we have the investors, and we have the consumers. And again, maybe there should have been a small dotted ring here saying food service customers or business-to-business customers. That is a driving force that we do believe may be one of those aspects that are not fully considered in the current picture that we're painting. So with that, it's really, to sum it all up, partnerships and collaboration. We do fully stand behind that, as other speakers have said. That is the way that we will bring this new, exciting, and hopefully very tasty products to the consumer. Thank you so much for listening.
Thank you so much, Anders. Very interesting talk. Now it's time for the Q&A. So it would be great if all the speakers could turn on their cameras. That would be wonderful. So we have a number of questions from the audience, but what I'd like to put to you all first is you've each outlined a step in the process of how we get novel foods to the plate. But how important are the links and the dialogues between all of the different players? I'm going to ask Muriel first, ladies first.
No, it's a great question because it's extremely important. Because when you start designing your process in the lab scale, that has implications for the production, it has implications for your dossier, for the consumer. And of course, that all needs to be circled back. So it's extremely important to communicate as early as possible when you are designing a process or a product, to think of what's coming up. Also the sustainability, customer acceptance, economic viability, all those things. So the entire chain is involved right from the start, I would say.
Malaus, what's your view on this?
I think that, it's a very exciting but also very complex field. And you see that we have tackled from different perspectives and many players are involved. So I think communication and engagement is a very, very important aspect. And from our side at EFSA, we pay a lot of effort and time and resources to be there, to communicate, to engage. And also there are more and more activities and platforms to interact with the industry, with academia, to help by giving pre-submission advice. But we think communication is it's something major, and it should be done among all the players in the field, especially when you develop something, when you prepare an application dossier, during the assessment, and also later at the marketing level. Because also, as it was highlighted by one of the previous speakers, consumers is also a key aspect of this process. And regardless, something is very, let's say, exciting, but it's safe. If in the end consumers are not into it, then it's something that needs to be further tackled.
Yes, exactly. Anders made a very good point about the nomenclature around what we're calling as an industry, precision fermentation, cultivated meat. But Anders, what's your opinion on what we can do better to communicate to end consumers about this novel technology or these novel technologies?
Yeah, it is the. There is no silver bullet, I guess. The consumer is more and more informed. That is one thing which is clear, and that goes. That's a global trend. So trying to fool the consumer is it's not going to happen. It will not fly. Now, to begin with, maybe the industry first needs to align and then communicate with the consumer, whereas doing it the other way around will probably be more confusing.
No, I completely agree. And when I talk about cultivated meat, I say cultivated, cell cultured. So yes, I think the terminology is not defined. And also, I don't think we're also hearing a lot of the media using very negative terms like lab-grown meat, in vitro meat, which is really not going to help convince consumers that these products are safe and they're actually foods. It sounds very alien, I would say. Timon, from your experience with setting up this code of practice for tastings, you mentioned that one of the objectives was to raise awareness. How did you measure the impact of that awareness?
Yeah, well, it's actually really hard to measure because there's of course two things. You have the media exposure these tasting sessions get, and that was enormous because every national media featured it a couple of times. So I think a lot of people in the Netherlands got a good view on television, in newspapers, in online articles of how these tasting sessions were, and also the pictures that accompanied it. So really, that really helps to draw these types of innovations out of the lab and really onto your plate, and that is a big factor in raising that awareness. And actually measuring how the Dutch audience sees cultured meat or other types of innovation, that's a bit harder to measure, of course. But maybe to complement Anders also, it's something we've worked on with behavioral experts when it comes to talking about these types of innovations. And it confirms what we saw, that it doesn't help to feature people in labs with white coats on, with complex machines in front of them, but really to show what the end product is, how that is really similar to what they are consuming already for decades or for centuries, actually, to really also show the outcomes from these tasting sessions, that these are very ordinary products, that they are intended to be that way, to make it as much as the same as possible without all the negative effects that go with it. So that to summarize. Yeah.
And from the tasting protocol that was implemented in the Netherlands, have other member states approached the Netherlands to learn more about how they went about it? And do you know of other member states that are working on similar protocols?
Yeah, we haven't heard officially about other member states really working on these types of protocols. We do know that our government has been asked about these tasting sessions and what could be learned from that. And we also got a lot of questions ourselves from other companies or other associations in other member states on, well, how did you approach this and what can we do to get this off the ground in our geographies? And we, of course, happy to share that. Yeah.
Yeah, I have heard that there are protocols being developed in Estonia and Lithuania. They're two regions that I've heard of, but nothing concrete yet, I don't think.
No.
And just another question on the tasting, because it's such an interesting topic. I think you did mention this during your talk, what the next steps are. But what everyone wants to know is this going to be opened up to non-Dutch companies, and will the scope be broadened to other novel foods, so not just cultivated meat? And if there's a timeline for that?
To begin with the last question, we would like to have tasting sessions for example, fermentation products as soon as possible, of course, in the Netherlands. And we're also actively working on that with companies from our membership to make that happen. In terms of the timing of those tasting sessions, we expect the Dutch government will not be working on facilitating these types of tasting sessions for products from fermentation before they have this evaluation of the cultured meat and fish pilot phase. We will urge members of parliaments to put the pressure on Dutch government to make that happen as soon as possible. But realistically, I think the earliest timing that tasting sessions could be possible, or that we will start working on them, maybe on the code of practice before the end of this year, if we get enough support of the members of parliament and with our ministries. But the actual tasting sessions taking place, that will take a bit longer after the evaluation, so after Q1 2025.
Okay.
And the first question I had was on companies from outside the Netherlands. Well, for the pilot phase, that will not happen because this has been set into the code of practice. We would like, of course, to make it possible to have also other companies joining in. But that's something that our government has been quite firm on to control the number of tasting sessions taking place, because the government does want to keep an eye on the safety, the controlled circumstances, the quality of the dossiers being filed to approve these tasting sessions. So my assumption is that this requirement will stay for a while, unfortunately. But I think our government does need to strike a balance there. So yeah.
Yeah, no, it's completely understandable. Yvonne Dommels has proposed, instead of having local protocols for tasting sessions, would it be possible to have an EU-wide protocol?
Yeah,
if
I would
not,
love that.
what can we do to solve it? I think talk to the European Commission about that.
Yeah, exactly. And I think it's actually more easy to develop national practices than the European one. That will take more time and efforts. But I would love to see that happen. And that's also something we can also work on, of course, through our national governments to call for such a initiative. But I also think when it comes to all kinds of novel foods applications, there's of course also a bit of a sensitive debate going on in Europe in member states right now. For example, when it comes to cultured meat. So I think it's also wise to be a bit careful there when it comes to bringing this up.
Absolutely. So we've discussed a lot about the need to have an optimal novel food approval process to be as efficient as possible. And there's a question from one of the attendees. So given the cost of getting a novel food authorized, what do you see as the biggest commercial advantage of going through the process? So what is the benefit of going through novel food approval in Europe? Maybe Ermelaus, you could start with that one.
Yes, of course. Yes, of course. As we said before, EFSA makes just a part of this process. And our risk assessment is, I would say, a very thorough one. And European citizens are used to one of the highest standards of food safety across the world. So if a company goes through this assessment and let's say gets the safety okay from EFSA, and then this product is marketed after the risk management approval in the EU market, I would assume that this could give the green light to a producer to place more easily their products in different markets if they have not already done so. And we have seen that many of our applicants, they are really keen on highlighting the positive assessment of EFSA on their websites when they market their products. So because European citizens are used to have this reassurance that what they eat is safe. And EFSA makes sure that these assessments are done using the most up-to-date science and in the most comprehensive way.
Any other of the panelists want to jump in here about the benefits of a novel food approval in the EU?
Well, maybe an obvious one is that the European market is quite a big one. In number of people, it's bigger than the US. So.
Yes, absolutely. And once you obtain a novel food approval, it's approved for all 27 member states and the other countries like Norway, Liechtenstein, Switzerland, etc. So it's, you have a lot of market access for that one approval, which is a huge benefit. And I think also it's worth noting the data protection that applicants can request when they submit their novel food application. And this basically grants 5 years data protection to the applicant for the data that they provide as part of their application. Now, this doesn't mean complete market exclusivity, but it means that that data is protected. And if another company wants to sell the same product, they would need to submit their own novel food dossier with their own data. So I think that's a big incentive as well. I don't know if anyone else has anything to add there. No. Okay, then we've got a question here. This is directed to Muriel. So thank you for your insightful talk. So how do you handle the scaling of continuous processes? And are there any challenges in transferring from laboratory scale to pilot scale and beyond?
Yeah. So we've run continuous, say, fermentations and downstream purifications up to our 15,000 liter scale. It is not more difficult than scaling up another process. I've also seen a question about the link with the downstream purification, but there it's actually the most of the purification steps are continuous. So when you run a fed batch or a batch process, it's actually a bit more difficult to pile up all the material and then start a continuous downstream purification step. But there are so many advantages to continuous processing. Once the process is running stable, it's much easier to follow up and to control than a fed batch or a batch-wise process. So we basically approach that in the very same way as we do other processes. We have a thorough preparation of the trials and look at where the risks and sensitive points could be. And we define backup plans for that. So we make sure we can keep on running when things run difficult, which is typically what you want to experience during piloting.
Brilliant. Thank you. And a question for Omelaus. What are the main challenges that you face when approaching the risk assessment of novel products, especially when these novel foods haven't been assessed before?
Thank you for the question. So actually, most of the novel food applications that we receive are products that we have not assessed before. And we have prepared the guidance that is an overarching guidance that provides guidance on how applicants can build up their, let's say, scientific approach to prepare their dossier, how to collect and assess the evidence, and which things to consider in order to decide if they should provide or not some additional evidence or conduct some additional studies. So you can understand that the novel foods, as we said before, are very heterogeneous. So we assess many different stuff. And of course, we have, in order to take care of this assessment, we have a multidisciplinary team of experts in the EFSA working with novel foods, also in the NDA panel, a panel on nutrition, novel foods, and food allergens. And also, this heterogeneous nature and needs for assessing novel foods are reflected internally at EFSA because we work all together with colleagues of different scientific backgrounds with different experience in order to be able to cover the vast array of novel foods arriving. I could highlight for sure that in some cases, I would say practical difficulty is the quality of the dossiers that we receive, because it's true that maybe the minimum required data are there to begin the assessment, but when we dig into this data, we find out that there are many data gaps that the applicants have not considered. There are many conclusions that are not supported by evidence provided. So, of course, this is something that we have to extensively assess, and in some cases, it can add up to the overtime of the assessment. So, I think that this new guidance that, let's say, consolidates the experience we have acquired so far by also seeing what the applicants perceive and how the applicants tackle their novel food dossiers should help minimize data gaps, and also allow the applicants to build better their strategy when preparing their dossiers.
Brilliant, thank you. And we have a question about the guidance, actually, about the updated guidance. So specifically around bioinformatics used as part of the allergenicity assessment. And the question is, you indicate that to continue with an IgE study if you find a hit with a known allergen, but what is the definition of a known allergen? Are those only the major allergens? As with bioinformatic analysis, you get a lot of hits with proteins to minor allergens as well, which are not clinically relevant.
Thank you again for the question. So indeed, this bioinformatic analysis can reveal many hits for, let's say, some of the major linked to some allergens that can be found in some of the major allergenic foods that are regulated. They are included in the list of the things that need to be labeled. Of course, we will assess data on other allergens that will be highlighted by this approach. But this does not necessarily mean that it will have an impact, let's say, on the safety conclusion. But in some cases, we have been asked by the risk managers to be more extensive when we provide feedback on the allergenic potential of a novel food. So our experts will assess this data, and we will report in the opinion the major findings, and then it's up to the risk managers to decide how they will deal further with information. For example, if they will need to take some further labeling measures with regards to a specific novel food, or if they will, let's say, disregard or give less importance to these findings because, for example, they could be similar to other products that are already in the market, that they are not subject to mandatory labeling, but they can still trigger allergic reactions to sensitive individuals. So just like overall, we prefer to have some more data to assess a more comprehensive body of evidence, and then to report the main findings in our assessments.
Brilliant, thank you. And there's one more question, or two more questions for you. I'll see if I can squeeze them in. So Camilla has asked, is any animal testing required for products from cellular agriculture? So I assume she's referring to the subchronic toxicity study in rats.
Thank you. So in one of the slides, I illustrated the tiered toxicity, ADME, and toxicity testing approach. And the toxicity testing approach comprises both genotoxicity testing and also repeat dosed animal testing. So if we go directly to the 90-day study, it is included in the first year, but if the applicant, considering the available evidence, argues that this study is not needed because there are other type of evidence that can, let's say, lift the need for conducting this study, then an applicant can proceed with submitting their dossier, and the experts will assess the reasoning provided that substantiates potentially the no need for such type of studies. And of course, this applies to all novel foods, including those from the so-called cellular agriculture. But there are provisions already in the guidance, also in the previous version, but also in the new versions highlighted, because we're trying to align our guidance with the efforts in the European Union to minimize and potentially in the future even phase out the animal studies for the safety assessment.
Super, thank you. And one more question, and this is relating to proposed uses and use levels, and whether EFSA will restrict the level of RNA from microbial proteins.
So actually, this was one of the issues that we have already faced in some of our risk assessment for novel foods produced with microorganisms. Currently, there is not an overarching limit, an overarching acceptable level for the intake of RNA or nucleic acids in general from a safety point of view. We're aware of some proposals that, let's say, have been made quite in the past by WHO about intake of nucleic acids from single-cell proteins. But yeah, currently there is not an overarching level. And if there is something that arises, like some evidence that arises during the assessments, then of course our experts will take this into consideration in order to decide if something more is needed to conduct further the assessment. But bottom line, no overarching limit for the intake of RNA from novel food intake.
Excellent, thank you. You were certainly put on the hot spot there. We're really close to running out of time, so I just wanted to wrap up this session. So thank you so much to all of our speakers. It's been incredibly informative, and thank you for sharing your insights.